So I will be talking about our work in Ladakh region in India. So this work was done in 2018, and what we did was mostly surveyed the Urial distribution in Ladakh, and we estimated population. So this is mostly focused on double observer survey, this workshop, but uh, what tell about this paper, this work is that how we went about doing the double observer from occupancy estimate, and how we did the both thing, like distribution and population density estimate of Ladakh Urial in our landscape. So Ladakh Urial is so Urial are found in West and Central Asian mountains, and they are dimorphic wild mountain sheep. And so basically they have these very similar features like other mountain ungulates, where males have larger horns and different features than females. And mostly Urial are found in arid and low productive ecosystem and near main river valleys. They prefer much more rolling mountains than other ungulates, most other ungulates, if you consider something like ibex and other ungulates, blue sheep. Urials prefer much more of a rolling mountains. Now their ranges are quite big. They are found from Iran to India, but they have like arguably they have 6 different subspecies. There are a lot of controversies around these. People don't agree and of the systematics, but you can see that the range of these Urial distributions are quite huge, like from Iran, Kazakhstan, Turkmenistan, like all these areas, Afghanistan, Pakistan, India. So it's quite widespread, but it's one of those most vulnerable wild ungulate at the same time. So what we have in India is the subspecies Ovis vignei, which is considered Ladakh Urial, and this is also a vulnerable ungulate. Now they are mostly distributed in India and Pakistan, mostly in Ladakh region and Gilgit-Baltistan in Pakistan. So now our studies show that they are mainly found in secondary river valleys, whereas like they face several threats like trophy hunting and habitat destruction, habitat degradation. So it's really important to understand their status. So we went on to do this survey because there are very few records and like with good survey techniques that can actually tell us what are the situations for Ladakh Urial. So what we decided to, so these 2 questions that we asked is, where are Ladakh Urials distributed in Ladakh region, and what factors drive their distribution? And also the second question was to know about the population status of Ladakh Urial. So to understand the distribution, we used occupancy framework, where presence and absence of the like target taxa are recorded, and it has a lot of different assumptions. I'll not go into details here, but there are assumptions that talks about temporal and spatial closure within a time frame. But at the same time, it also allows you to do temporal and spatial replicates. So basically what happens is that the presence and absence data tells you where animals are distributed at the end. So here we have this IUCN, existing IUCN map for Urial distribution in Ladakh. Now we use local knowledge survey based approach to understand, and also previous records, anecdotes, and we used our own ecological understanding of the species to actually delineate an area where Urials can be possibly found. And we have laid 5-kilometer trail on in those areas. Sorry, 5 into 5 kilometer grids in those areas. Now within each grid, we have these. We laid like 5 kilometer trails. Generally, it is done in the middle portion of the grid in one specific direction, such that trail represents the whole area. And then you can like before going to field also, you can actually break it into 5 sections of 1 kilometer replicates, which become a spatial replicates for occupancy. Now this is challenging in mountainous terrain because of these like straight trajectories that you can't take. So generally what can be done, you can walk a distance of 5 kilometers within that grid, which is representative of the grid, and then you can actually break it into again 5 sections after you come back from field. And so you walk these 5 kilometer distance, break it into 5 sections, and we recorded like all signs and direct sightings of Urials, but we discarded sign of Urial because that can be confusing because of the presence of other ungulates in the landscape. And if you think that some covariates that can be measured on field, you better collect those covariate data, because otherwise you come back from field and you can't collect those data in some cases, because these covariates will be really useful to understand which are the factors that are important in driving Urial distribution. Now we surveyed like total of 137 grids, which covers like 3,425 square kilometer area, which is kind of a huge area. We did not collect covariate data because we thought of some of the covariates that we can collect on ground are not representative of the grids, or they may not be very important for Urial distribution. So we rejected climate variables because the area that we sampled is very similar with its climatic covariates. So we rejected those. What we took was 4 covariates taken from Google, taken remotely from Google Earth Engine, using Google Earth Engine. And these all covariates are in a grid scale. So what we have done is taken mean elevation, variance of elevation, mean slope, and variance of slope. And then we use single season species occupancy framework. Now you can see the yellow grids are all grids which do not have any Urial presence. We did not record it in our data. And the green ones have Urial presence in them. We had 22 grids where Urials were present and 25 encounters in total. And detection probability was like 0.16 in this landscape. So basically it's quite a low detection probability. Now with occupancy estimate, what we found at 0.32. So basically 32% of the area are used by Urial. Now you can see there are a lot of variance that come variation in the occupancy estimate, and that can come from occupancy results, and you can see that you have a better picture of Urial distribution in Ladakh and also in Kargil region. One thing that you have to remember, that there is a considerable Urial site use possibility outside the IUCN map. So that is like inclusion of Urial range in that area. Now we see that like all those covariates have positive relations with it, but only elevation has, like, elevation explains 70% of the variation, whereas additive model of elevation and slope explain 28% of the variation. So what we understand is that the elevation is the most dominant factor in this landscape. But we'll have to remember that there is a physiological constraint in all the animals. They will not be found beyond a limit, elevational limit where physiological constraints are imposed. So now we'll go into this population status work, and we use the double observer survey method. And as we all know now that it is one method where we use two observers, and this is actually a capture recapture method, whereas where capture like detection by one individual is a recapture by another individual. So and it also assume a temporal and spatial closure, and you guys will have you guys will know that these are the assumptions Muni must have told you guys earlier. So I'll not go into the details of those assumptions then, but what we have done, we have actually taken these blue sections that you are seeing, which we considered as the prime Urial habitat, and we have selected those two sites. One is Heniskot Lamayuru Takmachik landscape, or HLTL, and another is Aparsham landscape, or USL. So here are these two landscapes, which are very similar in its topography and all, but there are other difference in local culture, local religious practice, and there are other factors, presence of army personnel and other kind of factors that are little different. And also Upper Sham landscape has much more nomadic herders than HLTL landscape, whereas the HLTL sees a lot of crop production in the landscape. Now what we have done, we have selected one site and broken them into survey blocks. And what we have done is we have used ridgelines, valleys, which we think fulfills the assumptions of this spatial closure, and these prominent features were used to create these blocks. And we have to always remember that this should be ecologically relevant and logistically possible. Now within the block, we have laid transects, and we have to remember that for double observer survey, if you can't lay a transect beforehand, like in QGIS or Google Earth, something like that, if you can't do it remotely, at least you'll have to reach the site and decide on which ways to follow. Because if two observers follow different paths, then a lot of the assumptions fall apart. So we'll have to do that. So we laid those transects, and we kept like basically we kept 20 meters 20-minute distance between observer 1 and observer 2, and we walked those transects. So in HLTL, we covered almost 345 square kilometer, and USL we covered 280 square kilometer. And we counted animals. We tried to classify them according to age and sex, because that also helps them helps us identify how age and sex structures are in these populations. So our result at the end shows that HLTL has less. So HLTL population size is less, and USL it is more. Now here we have capped the lower confidence interval as the minimum count. So yeah, that's why this is like the same. Now, if you look at the density, USL has considerably higher density estimate. In most of the Ladakh Urial work that was done before, which is very few, but at least 3-4 works that were done before, the densities are always lower than one individual in most of the areas. But Upper Sham landscape has like 1.27, whereas HLTL has like 0.996, which is very close to 1. But if you look at this mean group size, it's quite interesting to look at this. We see that HLTL has a higher group size, mean group size compared to USL. And one of the things, so one important factor is that the HLTL group size are quite big. It's more than 100 sometimes. So it's quite big, whereas in USL we have more number of herds, but the individuals in the herd is much lower than the HLTL. So that also gives us insight about some ideas how the populations are structured. Where you can always say like USL are clumped, whereas HLTL are congregated. Now, if we in last section, it's a very simple work and simple use of 2 methods. But what we achieved is that a larger area survey than what we used to know from before. Urial distributions are larger, but it is still limited in most of the areas. You could see that the amount of yellow among that 137 grids were much higher than the green in the map. So basically it's still limited, the range. Population is close to 700. There will be little more than this if we include other parts, but this is actually really helpful to understand, because this is actually the prime habitat and it will be very small departure from the actual truth. Now we need conservation work in this landscape, but we need further research and long-term monitoring of population status using double observer survey and other methods. So yeah, so that's mostly it. Thank you. So I acknowledge Forest Department for helping us in this work. The work was done in collaboration with Nature Conservation Foundation and Forest Department. So I thank them, and these are the people. Some of them started the work, and some of them helped in analyzing data. And Karmaji, Dorje, Lobjang, Spaldon, who are like amazing in field. So a big thanks to them. And I will like to have some questions if you guys have.
Super. Thanks so much, Abhirup, especially for really giving us a great sense of how, we can use mixed methods really to understand species distributions and sort of at larger areas, and then use that to really, direct population assessment surveys for smaller areas, because obviously doing that over larger areas is not always feasible. So yes, guys, I think if people have questions, feel free to ask or type in the chat, and then we'll wrap up the sessions, both for Abhirup or yes.
Generally.
Yes, or generally. So there's some comments coming in. As far as not yet included issue. Yes, Stefan. Actually, that was one of the hopes. I think you've been talking to Dr. Bhatnagar, right? So he was also part of this effort, and I think the hope is to try and.
Yeah. Actually, Yashvir had provided me with quite some information, so I'm not sure if we not yet included this information. I think we must have considered it in some way. I don't know if there is a more recent publication. Of course, I would be interested, because I'm, really enthusiastic about Urial, which I consider the most overlooked mountain ungulate, which is totally ignored, and often people don't know it. Even local people don't know it, and it's always everybody talks about Argali and Marco Polo sheep, and Urial are overlooked. Yeah. Just one minor comment for you, that actually, Savartsov sheep is now considered being an Agali in Uzbekistan. So by chromosomes and by morphology, it is a small Agali, not a Urial, which makes actually the Urial population a bit smaller even. But really great work and very interesting. And what I found interesting is that group size seemed to be not correlated with overall population density, which is maybe because the population densities did not differ so much between the two study areas. But what I noticed is that generally group size in Urial and also in other wild sheep it goes down when the populations are very small. So, and while large populations typically, or more dense populations, have a larger group size of maybe 50, 100 animals, which you cannot observe in areas with small populations. Thank you.
Thanks, Stefan. That's quite like interesting thing. Yeah. So we don't know actually the drivers of this group size in these landscapes, but there can be several factors. So not sure. We'll have to remember these things when we consider discussing the group size in later stages. Thanks.
Yeah, like, I mean, just a quick point. We've had a quick back and forth about this as well. You know, some thoughts are maybe group size is driven by some sort of hunting or, you know, other persecutions. But, you know, it's really hard to say that as well because we don't have the data as Abhirup said. But I think this shows you the value of things like Double Observer, because rather than just getting a density number, you know, you get all these insights that we can. Yeah. Yes, we will, Stefan. It's in review right now. So
yeah.
Hopefully, if it gets, and when it gets, when rather than if it gets accepted, we'll share. Okay. So any other questions for Abhirup? I'll just wait for a minute and then I can wrap up because I'm really sorry. I know we've been over time today. But yes.
Raki will be after you, Munip. I'm
sorry.
It's related to time. But it's fine. It's
I'm sorry.
It was great to hear from Abhirup. Thank you for doing that.
It is excellent.
So I'll just do, and if there are questions, please type in. I'll just do one quick slide of summary so we all leave with some material from this. So what have we learned from today's class? You know, mountain ungulate study areas doesn't, when you define a study area, there isn't one fit all strategy. You know, for double observer study areas, you have to further divide them into survey blocks and then the transect. Reconciling data between the two observers is key for the capture-recapture nature, as we've discussed. And studying ungulates over large landscapes can be achieved through mixed methods, and as we explained, through occupancy and the double observer method. And just lastly, next class, we'll go a little bit into the analyzing the data. So if you guys have your R Studio with you, it'll be great. And we'll go a little bit into the importance of 8-sex classification a bit more. And lastly, into studying other ungulates, which I promised we'll do this time, but just did not have time to put it in, but like things like muscle and things like that. So that's all for today's class, and I will stop sharing. And yes, there's some comments. Abhirup, people are asking if the report can be shared. I guess we can share the report, right? Because that's similar to our publication.
Yeah, we can share the report, I guess.
Sure.
Yeah, but yeah, we can discuss that later because.
Yes,
yeah,
I'll have
but you can share it.
Cool. Okay, so if there aren't any other questions, then I would like to finish the presentation after each session. Yes, Tashi, I will do it right after this. I know I was very late for the first one. I will do it right after.
Munip, can I ask, make a request on the part of SLN? Can we ask everyone, we did this for the snow leopard training.
Yeah.
I would like everyone to maybe put on their camera just for a moment, just to show the diversity. If you're okay, you don't have to. And then, I don't know, Munip has this great impressioning of an ibex or an agali, and you can choose your species of interest. So, and we'll just take a second and we'll just take a group photo all together. It's great seeing Wanjun with your son. Nice to see. Oh my God, so many sons there. So if you want to join in the group picture, please turn on your video just for fun, you know, just to show so many to thank you all from coming from so many different places of the world. It's great to see so many faces.
Yeah, really, actually, while people won't switch on the cameras, I really appreciate everyone coming in. I mean, it's a long time, 2 hours. It's almost 2.5 hours now. So thank you very much. But yeah.
Okay. So I think I'm gonna take a screenshot of. It's in 2 pages. I think we've got 2 pages of faces. So I think I have to do a little video. So I'm going to do a video here.
Are we
maybe
to be
Munib, you can lead us through that. Maybe Munib?
So guys, let's try to be ibex or ibexes so we can sort of make a little, horns on the top, but our faces in the front, so Justine sees it. I think Abhirup is going to kill me later.
Great. Okay, I'm going to the second page with all the ibex. Thank you all for joining. Thank you. I think I got it. Thank you, Munib.
Cool. Okay, guys.
Thank you.
I will share the presentation next, and yes, we can chat over the Google Groups if you need anything. And see you next week.
Yeah, thank you.
Okay.
Cheers, guys.
Bye, everyone.
Bye. Take care.
Bye, everyone.
Bye.